An ideal day, I think, in my life as a PhD student in the CSD program would involve some sort of live imaging, because I love that. It is the coolest thing to have like live neurons on the microscope and see things moving in them. I find that to be very exciting. But then also, if that could correspond to a day that you finished your analysis of something and have come up with like a concrete piece of data that shows that like, oh my gosh, my hypothesis is supported by this data, that is super exciting. Being able to share that, if it also corresponded with like lab meeting, where you're meeting with your peers and you can share the amazing data that you just discovered with them, oh, that would be like the best day.
I'm able to have some coffee in the morning.
That's a great start.
I think any day where I'm allowed to just like have a nice mix of interaction with my lab, interaction with my mentor is always a really great really great thing. Being able to get in and do some experiments, even if it's like I don't get great results. I really do like sitting down at the bench and kind of just working hard at a goal. That's a good day. Yeah.
Honestly, my favorite part is that I get to solve puzzles all day, and I get to think of puzzles that no one's ever thought of and figure out how to solve them. And I think that actually is what makes for the best day. It's when you come in the morning with this question and you think for a long time about how to solve it and you kind of get stuck, and then you go to a seminar and you get a piece of the answer, hearing it from someone, and then later in the afternoon you go to lab meeting and you kind of like share these things with other people, and it's through this communication that you're able to like put together these little pieces and then finally design this final experiment. And if you're lucky, it turns out the way you thought, and if not, you learn something even better.
So what lights me up, in lab is when I find some new piece of data, some new image, and then I get to talk about it to my colleagues, and we come up with all of these hypotheses about what could be happening and how something works. And sometimes we'll go on for half an hour, an hour, just talking about all these ideas. We'll bounce ideas off each other. Honestly, that is the most fun thing I experience almost anywhere, anytime.
My favorite days in lab are when I get to be maybe sitting on the microscope after doing this experiment for probably at least a week, getting different time points and sectioning, and then finally doing this like final imaging project, and I see something cool, something I haven't seen before, and I get to go into Lori's office and show her my computer and show her the pictures I just found, and her just being really excited with me, and then I get to go tell everyone in the lab. I think, yeah, those are the absolutely most fun days that I have in the lab.
So a really exciting day that just happened recently actually was we had been working on this project for a really long time, trying to purify this protein, and we kind of I nervously came into lab, at least all of us were a little bit on edge with whether or not it would actually work. And then when we got those positive results, it was just great because as a whole lab, we really come together. I feel like we support ourselves here, or support each other so much here. And that definitely was the case when we got this exciting result. Everyone was happy, including even the surrounding labs got in on it and were excited about it. And I think it's really just that kind of community support that makes a great day.
So a good day for me would be, working in the brain development lab, I study synapse development at different time points in the mammalian brain, and I use the mouse as a model. And mouse development happens over the course of, say, 2-3 weeks. So you plan experiments, you wait the 2-3 weeks, you do your imaging, and you hope that your results validate your initial hypothesis. And obviously there are moments where it's quite the opposite, but when you get results that do validate your personal hypothesis and you see it right there on the confocal microscope, you're just like, you just get this feeling of excitement where you're just like, yes, I did it. I got this piece of data. What can I, what does it really mean? And what can I do knowing this piece of information, and how does this contribute ultimately to synapse development? So to me, that's, gaining that useful piece of information to help sort of put together this idea of how a neuron develops in the brain is just an exciting thing for me.
A good day for me is when I get to share in someone else's success. I still like doing research at the bench, and I try to do it when I can, but I think it's become clear to me that the most rewarding thing in my job is to learn about someone else's experiment, and that they've been working really hard at it, and that it's been successful and they've learned something from it. And to share in that enthusiasm, and then think about the sort of wonder of what the next step is and get excited about new possibilities is really the greatest joy of being a scientist, and I'm fortunate to be able to do that all the time.